Okay, good evening, everyone. I'm Yadvinder Mali from the School of Geography and the Environment, and I'll be hosting a speaker this evening. Is the mic okay? Can you hear me? Is that okay? Yep. Okay, good. I'm Yadvinder Mali from the School of Geography and the Environment, and I'll be hosting a speaker this evening. And before we start, I want to remind you of a couple of things. Firstly, this lecture is being filmed and live webcast, so you should be aware of that. And afterwards, next door, there is a drinks reception to which all of you are welcome to come and have a glass of wine and an informal chat with the speaker and with each other. So to introduce Brian, Brian's here, has been here since January as an Oxford Martin School visiting fellow. And Brian did his PhD in the 1990s with Jim Brown, who was the father of one of the key figures of macroecology. And his career since then has been marked by a number of features. One is an impressive volume of papers in science and nature, which is an enviable quantity. But far more importantly, his work has been characterized with a vast, sometimes breathtaking scope and vision of the scale at which he thinks and the breadth of the concepts with which he engages. Sometimes these concepts have been controversial and contested, but they've always helped to define the field, move the field forward in testing some of his provocative ideas. And my own experience with Brian goes back a few years where we've been working together, and his way at looking at data has made me look at my own data in different ways and look at different questions and a larger scale of vision in the data that we have. And Brian's talk today promises from its title to be of similar grand scope and vision, and I look forward to us all hearing it. So Brian Enquist, welcome.
Thank you, Yadvinder, for that very nice introduction. But I should say and pause that if I can turn the spotlight back on Yadvinder, Yadvinder was just elected as a fellow to the Royal Society. It's arguably the most prestigious scientific society in the world, and we're all lucky to not only know you, but to have you here at Oxford. So congratulations to Yadvinder. Okay, so in researching my talk, I pretty much knew what I wanted to say, but I wanted to kind of scroll through the bowels of the internet to see what else I could find, and I stumbled upon this very interesting website. This is www.humanlongevity.com. And at humanlongevity.com, they promise you a future that's going to be very bright indeed. One where all the diseases associated with aging are not only cured, but also changing the way medicine is practiced. Okay? Longevity.com. So, with that in mind, let's step back and think about the natural world. Let's step back and think about biology. Step back and think about the diversity of life. Life is amazing and complex, right? And this is reflected in the different animals and plants and various things that we can measure in biology. But what I would like to argue today is that despite this diversity, despite these differences between organisms, despite the differences in how they interact with each other, that there are underlying unities of life. Okay? And these unities of life have actually been known more or less for a quite long time, since the beginnings of biology. Okay? And this unity of life is actually reflected in what I'm going to call biological time. So, the diversity of life. And let's think about time, in particular, how long things live, right? So we can think about microbes, lifespans of minutes to hours. There's a mayfly, lives about 24 hours. A nice drone ant lives on the order of about 3 weeks. House mice, about a year, if they're lucky. Domestic rabbit, 8 to 12 years. Put in captivity, maybe a little bit more. But the world record of animals in terms of living is this creature right here. This is the bowhead whale, lives approximately 211 years old. But in terms of living things, there's things that actually live longer than this. So this is the General Sherman tree in California. This is the giant sequoia, estimated to be approximately 2,500 years old, this one individual. Organisms can actually live longer than this. In fact, for a single individual, in terms of maximum lifespan, unfortunately, it's not living today because, unfortunately, it was cut down. And in the process of cutting it down and counting the tree rings, they discovered that it was the oldest tree ever recorded, Methuselah, 5,065 years old. Oldest known living non-clonal organism. Okay, but what about us? This is Jean-Louise Clement, okay, oldest living human ever recorded. She was 122 years old and 164 days. When you get that old, these days are important, right? She was born in 1875, 14 years before the Eiffel Tower was constructed. She used to sell painting canvases to Van Gogh. She died in 1997. She started smoking at the age of 16, and she quit when she was 117. She smoked not only 1 or 2 cigarettes a day, but no more than that. And she also had a glass of port every day. The secret of her longevity, she says, was eating chocolate, drinking port, and rubbing olive oil over her skin. 122 years. So time, right? Well, time is what a clock reads, right? To know the time, you go to the clock. To watch the passage of time, you watch the clock. Time passes at a constant rate. In terms of the history of science, we can really go back and characterize the history of science in terms of our abilities to characterize and measure time. Scientific progress is really reflected by this continuous improvement of measuring time. From things like Stonehenge to these early mechanical clocks, such as the one in Prague in the Czech Republic. The uncertainty of these early clocks spanned on the time order of maybe days or hours, up until modern day with our modern atomic clocks. Our uncertainty in measuring time now is 1 second in 30 million years. That's how accurate our timekeeping is now. So these clocks all tick along at the same time scale. But this ticking of the clock, I'd like to emphasize, is on a time scale relative to how we perceive the passage of time. Right. So good is our ability to characterize time. We can go back in time and characterize very accurately with many different methods the geological time scale. And we can place this geological time scale in the context of biology. So we know approximately the Earth is 4.8 billion years old. We know that life arose approximately 3.5 billion years ago. But life really didn't take off. That is, the diversity of forms that we see all around us until the Precambrian, approximately 541 million years ago. Okay. And during this time, this is where the major clades arose in terms of what we see today, in terms of the diversity of life. So, unlike our timekeeping with clocks, what I would like to emphasize today is that biological time is different. Biological time is relative. So let's think about taking a walk in a forest, right? Pleasant day, you go through, you see the breeze. Well, you hear the breeze, you feel the breeze on your neck, you see the leaves moving in the wind. But if you watch the trees, besides bending in the wind, they don't seem to do much. So what if we had the ability to take something like a forest and speed up human time? Basically compress human time of 1 year, compress it down to 30 seconds, and take that same forest and scale the time scale of a forest so that human time and forest time merges. So let's alter the temporal scale of a forest. Okay? And this slow biology that we see in the forest appears to come alive. So I stole a very cool PBS special that I saw about a year ago. This is The Life of Plants. And so what I'm going to show you is a time-lapse recording where they move then a camera then through a forest on the time scale of about a growing season, about a year, and you're going to see plants come alive, growing at the speed at which we would perceive them if they lived and operated on human time. Flowers grow almost instantaneously. Moss shimmers in the light. We see branches slowly start extending. Flowers are born, then they die. Seeds come to life relatively quickly. Plants begin to explore the light environment, moving their branches up and down. Vines seem to spin in the air as they search for various places to go. And fungi seem to grow quite rapidly. So increasingly, we're able to view biology on the timescales that we perceive time. So does biological time tick or pass at a constant rate? So what I'm going to argue is that no, it doesn't. Life's perception of time is different than geological time, but biological time does have constants. And it's these constants that we're just starting to understand, but they also have a long history in biology. And to boil down these biological constants, we don't understand where they come from. To make sense of biological time, we need to understand something about scaling. So to understand the dimensions of biological time actually requires 5 different insights. Okay, so let's move through these 5 different insights. Okay, first, let me give you a quick scaling primer in one slide. So our understanding of scaling actually goes back to Galileo in 1638. And so many of you already know basics of scaling, basic math you take in primary school. So if we want to take a simple Euclidean object, such as a cube, and blow it up in size, we actually know how the volume and how the areas and the surface areas then will then change as we go from small then to large. We know that the area then of a cube is going to be its length squared, and its volume is going to be its length cubed. And so Galileo actually effectively not only went back to these principles of similitude, but he realized that within biology, if you take the bone of a small mouse and a bone of an elephant, and you scale them appropriately so that they're plotted on the same size scale, what you can tell is that gravity is more important for the elephant than it is for the mouse. Because of gravity, bones have to scale differently than what you would expect from the simple scaling of a cube or a sphere or so on. And so Galileo used this principle of scaling to infer deeper insights into biology. Okay. So the second then insight I would like to pass along is that biological rates scale. Biological time scale. So this is the animal physiologist Max Kleiber, 1938. And one of Max's claims of fame in terms of one of the most important things that he discovered was how the metabolic rate of an animal changes as it changes in size. And actually, this is one of my favorite biological graphs that I like to teach to my undergraduates, because this is one of the only graphs that I know of where you actually have a man and a woman plotted on the same plot together. Now, we've come a long ways phylogenetically in terms of how we understand these relationships, but here we have things like mice, small birds, elephants, horses, man and women, all together on the same plot together. This is called the mouse to elephant curve, okay? So early on, what Max Kleiber noted is that as you go from small sizes to large sizes, the metabolic rate, that is how much energy an animal needs to live, increases with its size. But the other thing that he noticed was that it wasn't just a simple straight line. What he noted was that the slope of that line is actually 3/4 or 0.75. And at the time, that was very interesting because it went totally against what most people in biology thought that this relationship should look like. If you double the size of the animal, do you double the amount of food that you give it? Rhetorical question. The answer is no, because this dotted line is a one-to-one line. That's the doubling line. This other dotted line, this blue dotted line here, is the 2/3 line. That is, if the amount of energy an animal needs is proportional to its surface area, remember the example of a cube going from small cubes to large cubes? If your metabolism is proportional to your surface area of your body, we would expect the surface rule to have a different slope of 2/3. And over again, this launched a huge debate in biology. Why does biological rates and times scale with a slope or an exponent that's different from the 2/3 or that similitude rule from Galileo? So, biology isn't simple Euclidean geometry. Now, we can fast forward many decades, many years, and what we begin to identify is that there appears to be more universal biological scaling functions. So can we take the diversity of life and make sense in terms of how life works in a scaling framework? So let's look at this same graph again. This is body mass, small size, large size. And we're going to plot a measure of growth rate, how fast an organism grows. So let's put some insects on the graph. Let's put some birds also on the graph. Let's put some fish, some plants, some mammals, some protists, some zooplankton. Wouldn't it be great if you could put some dinosaurs on there too? Well, actually, it turns out if you cut open a dinosaur bone, you actually see rings like a tree ring in a dinosaur bone. All right, and actually, in a lot of reptiles today, you can count how old they are based on the rings then within their bones. And it turns out when you do that, based on some other biological knowledge, we can get an estimate of the growth rate of dinosaurs, and there they are. So all organisms appear to be described by an approximate scaling function. And the slope is approximately 3/4, and not 2/3. So what does the 3/4 or the 7/5 scaling relationship mean? Well, if we do some quick math, so this is metabolism mass to the 3/4. If I look at the metabolism per unit mass, how much energy it takes to support a given mass of animal, what you actually find is that as you go from small things to large things, the metabolic rate per cell decreases. A small organism has an incredible metabolic rate in its cell, whereas a large animal, such as an elephant, has a much lower metabolic rate. We see these relationships almost everywhere we look. This is heart rate, going from small mammals to big ones, number of beats per minute, how fast your heart is beating. Look at a whale, 20 beats per minute. We can go up to a human, about 70 beats per minute. Look at a shrew, smallest mammal, 600 beats per minute. If you've ever held a shrew, it's like they're vibrating. Mutation rate, how fast new bits of information are entered into the genome. As we look at small organisms to large, small organisms have a higher mutation rate, larger organisms have a lower mutation rate. Okay, so the third bit of insight, biological times scale. So across biology, biological times scale as approximately their size to the 1/4 power. Any timing, critical life timing that you can measure, ranging from body cycles, such as respiratory cycle, how fast your muscles contract, your cardiac cycle, physiological times, your circulation of blood, how fast it takes you to clear insulin, the plasma half-life of given different metabolites, your metabolism of fat, growth and reproductive times, how long it takes then for an organism of a given size to reach reproductive age, but also ultimately your lifespan and ecology. So this is a summary of many different birds. The slopes of these different relationships are all clustering around 1/4. So biological time is warped. The bigger you are, the longer it takes to do things. So this is a fun little bit of biology. So let's think about pregnancy in terms of how long different animals it takes for them to give birth. Okay, so I'm showing you how long some great pictures of a developing zygote here of an elephant, okay, small mouse. If we plot then differences in body size in these different organisms versus gestation period, how long then mom is pregnant, right? It scales as mass to the 1/4. Okay. Larger organisms take longer to give birth. But what's interesting is that as then the developing fetus is growing within mom, the metabolic rate of the youngster, while it's inside mom, its metabolic rate goes as the metabolic rate of mom. But as soon as you're born, your metabolic rate increases appropriate to the size at which you are born. So at birth, metabolic rate then scales like any other mammal of a given size. So biological rates and times scale. Biological time changes. It's as if biological time is warped. Okay? Salvador looks on. So why is this? Why do larger organisms live longer? Why do small ones live such a short time? So is this just numerology, this 3/4, this 1/4, and so on? Well, it actually turns out that there are a lot of different quarter powers in biology. 1/12, 3/8, 3/4, and 1/4. We seem to see these in many different organisms. And so this suggests that there may be something deeper. There may be some deeper biological truths. In particular, it suggests that deep down biological time is regulated or controlled in some sort of way. Okay, so remember, 5 bits of insight that I would like to pass along. What's the 4th one? The 4th one actually comes from trying to understand why we see such variation in biological time, and that is having to do with networks. So earlier, with work from Jeffrey West, who's actually a trained physicist at Los Alamos National Labs, and at the time, my advisor, Jim Brown at the University of New Mexico, we started not only pondering these different patterns in biology, but we wanted to know whether or not we could construct an argument for why we see these prominent patterns in biology. And here's the assumption, is that even you, as you're sitting there, if we dissolved away your skin, you are a sitting network. In fact, most of biology is a network, a vascular network, a network of pipes and plumbing that not only disperses resources through the body, but also transfers information. And an assumption of this work is that these biological scaling laws originate in the size and the geometry of these hierarchical tree-like networks. Okay? So if you strip away your skin and you compare your vascular network to a tree, mathematically, networks of plants and animals are remarkably similar to each other. But we've actually known this for a while. In fact, it was Leonardo da Vinci who, after a series of different paintings of landscapes, realized that there was an easy way to paint a tree. He called it the area preservation rule. And so from Leonardo's notebooks, we can work out how he actually worked it out, but what he discovered was that as you follow a tree from the base of a tree all the way to the tips then of the tree, this network is area preserving. That is, if we calculate the cross-sectional area of the mother branch here at the base, and we calculate the cross-sectional area of all the twigs at the tip, the area is preserved. It's the same cross-sectional area. So if you want to know the cross-sectional area of a trunk of a tree, count up the cross-sectional area of all the tips of the terminal branches. We've done that. I wouldn't pass along that fate to anyone. It's a hard work, but in general, area preservation seems to be the rule. So, networks, quarter power scaling. The first hypothesis is that cellular metabolism is a unifying measure of life. We can measure metabolism, not only the individual scales, but also at scales of the ecosystem. Metabolism controls the pace, form, and diversity of life. But what controls the pace of cellular metabolism? Well, ultimately, you can drill down to your cells. Remember your basic biology? Well, the mitochondria here is ultimately responsible for the flux of energy then through the cell. In particular, we can drill down even further to cytochrome oxidase, which is remarkably similar across life. But there's a bigger and important problem here. If ultimately metabolism then is being done within mitochondria, and thanks to the cytochrome oxidase molecule, a problem then is, how do we get resources to fuel your cells? Right? How do we supply all of your cells within your body? So let's use an analogy. Right? How do we design an optimal network? Right? So think of all of us living in a congested town, and we want to optimally satisfy the needs of all people. You want to get from one place to another as fast as you can, but because we all pay taxes, we want to invest in our infrastructure at minimum in order to maximize then the goals of everyone who lives within your network. So natural selection, we argue, has done something similar. And that hypothesis is that these quarter-power exponents reflect the constraints of how organisms optimally supply resources through these networks to supply their metabolic machinery. In particular, natural selection, strong natural selection to optimize biological networks. These networks maximally supply resources from the environment to all metabolizing cells, but yet minimize the cost in terms of resistance, in terms of transport time, of the transport through the networks. And how does an optimal network scale? As you grow from baby to an adult, the total number of cells in your body increases as mass to the power 1. That is, you double your size, you double the number of cells that you have. But in a big network, you need more time to deliver resources because the network can only supply, an optimal network can only supply resources at mass to the 3/4 rate. The demand is potentially greater than the supply, and because of which you have to turn down your metabolism in order for the organism to function. And I can give you a quick, simple analogy to maybe prove my point. So let's think about the metabolic potential of a mammalian cell. It's this, right? Incredibly fast, can run at high speed, and if you're a shrew, that's probably close to what's real. Now let's take that same cell in quotes, and put it into a big mammal. You're not going to be running very fast, right? You're going to have to idle quite a bit, but your time is slowed, okay? Because you're part of a larger network. Now that same cell, but placed within a network with few limits, open highway. Do we have proof that in general this networks constrain how fast metabolism, how fast life runs? So here's an interesting experiment that has been done. This is the metabolic rate per cell. That is the total amount of energy it takes then to maintain a given cell or a given volume of mass. As you increase body mass, normal living organisms then, metabolism decreases. But in these red cells here, these are cells then that are grown in the laboratory. That is, they're cells taken from these organisms of a given size and grown in a petri dish in the laboratory. And one of the first things that happens when you take a cell then out of an elephant or a larger mammal is that the metabolic rate of cells increase. So cells raised in the laboratory, free from the network, increase their metabolism. And their metabolic rate then is approximately flat, and it intersects the real world line here, and that size is the size of about a shrew, the smallest mammal. Okay? So this is consistent with the prediction that the constraints then of the network limits the metabolic potential of cells. So there's a shrew sitting right there, probably fluxing energy as fast as possible from a mammalian cell. So variation in scaling of biological rates and times is also constrained by what we call constants of life. And this is the last insight that I'd like to pass along. And so I'm going to give you a few different examples, but I'm going to give you one example, in fact, something that we use quite a bit in our current research. And we're going to focus on leaf diversity. Okay, leaves on plants, right? Leaves are really cool because they not only come in all these different shapes, right? But, yes, different colors of green, but leaves are found, obviously, in a lot of different angiosperms and gymnosperms all across the globe. And ultimately, leaves are responsible for how much carbon then comes into ecosystems, but how much sunlight then, the energy from sunlight is ultimately passed along to then fuel the biosphere. So leaf diversity. Leaves are pretty interesting because if we look at the lifespan then of different leaves, I should say that these are little cocktail swords. So this is the ecologist in the field marking individual plants and studying then their life cycles, but also monitoring how long these different then leaves of, in this case, desert annuals then live. In desert annuals, their life cycles are timed to these short little durations of rainfall that occurs. These plants grow up in size, make use of the little bit of ephemeral rain, and then once the dryness of the desert sets back in, they quickly then die, but then reproduce before doing so. These leaves last on the timescale of a few days to weeks. And in starting this work, I started to realize, well, leaves, there's some variation in leaf span, but actually there's an enormous variation in how long a given leaf can live. So here's an araucaria tree. Leaves on araucaria actually can live on the order of about 25 years. And it's only been on the time scale of about a decade or so that research in terms of how long leaves live or plants live has really started to take off. In fact, we don't really know much about how long plants live, especially some very interesting ones. And probably the longest lived leaf is actually from this plant. So this is Welwitschia, grows in the deserts of Namibia. When it germinates, it basically produces just a few leaves, puts them out, and looks like it's vomiting this green mass. Looks quite disheveled, but this is the normal state of Welwitschia. These leaves can live on the order of about 1,000 years. So let's think of a leaf as a fundamental metabolic unit. Again, a fundamental unit then responsible for fixing energy from the sun and powering then our biosphere. So again, leaves dominate carbon, water, and nutrient fluxes of terrestrial ecosystems. But what I'm going to tell you is I'm going to strip away this thin veneer on the outside of a leaf and peer in, because underneath the greenness then of a leaf is a wonderful and beautiful network. And this network and the various traits of leaves associated with the flux of energy and matter are honed to match the local environment. So this is an aspen leaf stripped away of everything on the outside to reveal the network itself. Right? So biology is a network. That network then is responsible for, say, all of the growth or production that occurs within a tropical forest, ultimately through leaves, through these networks. And so part of the work in collaboration with the Yadvinder is that we have been sampling different leaves in many different environments. Here we are working along an elevational gradient in the Andes of Peru, going from the Amazon to tree line, to understand the diversity of how plants function. And one of the questions is, how can we understand the diversity of branching of both trees, but also within these networks of leaves? Because we think these networks inform us in terms of how these forests not only function, but what will happen in the future with climate change. So can we predict how climate influences the venation diversity that we see in these leaves? And so these venation networks are actually quite beautiful. So I'm showing you several different examples of several different species. And if you look closely, you can actually start to tell differences between species in terms of their different networks. So this diversity of leaves that we see can be characterized by what we call the leaf economic spectrum. Okay? So we can collapse all of the diversity of leaf form and function along this economic spectrum. And so this spectrum consists of how much carbon it costs to build a leaf, how long it takes to pay back the cost of building that leaf, but also the lifespan of a leaf. And so leaves can be kind of anchored on two ends of the spectrum. Right? We can have leaves that cost relatively little in terms of creating. That is, it's a cheap leaf. But these cheap leaves tend to live a very short amount of time, and the return rate, how much time it takes to pay back the cost of creating that leaf, is a short amount of time. So let's then compare that with the opposite end, where we have leaves that live a long time, cost a lot of carbon to make, and the return then rate, right, is also scaled in accordance. And in general, we have leaves that either high carbon cost, long life, low carbon return rate, or low carbon cost, short-lived lives, and high carbon return rates. And if we measure many different attributes of leaves, such as how much nitrogen is in them, how much carbon then is in them, how long they live, how fast the rate of photosynthesis, how fast they take up carbon then from the atmosphere, and then we plot our predicted relationships according to this fast-slow continuum based on their networks, we can begin to describe these different variations in biological time of leaves. And if I were to compress all of this down into a single axis, we have slow leaves that live a relatively long life and fast leaves that live a relatively short life. You can tell that by looking at their networks. Right? So effectively, we have leaves that are the James Dean of life, live fast and die young, and leaves that live slowly and steady, but ultimately, at the end of the day, end up making up the difference. And what's absolutely remarkable about the diversity of leaves across the planet is that for all leaves, it takes approximately 4 grams of carbon that's assimilated per 1 gram of carbon invested in the leaf. So over the lifespan of a short-lived leaf or a long-lived leaf, all leaves are approximately assimilating 4 grams of carbon for every 1 gram invested in a leaf. A constant. Why is this an approximate constant? Why is it 4 grams per every 1 gram invested in making a leaf? Well, we don't know. But it's clear that you can take those 4 grams, at which a leaf is going to then assimilate over its lifespan, and pack it all in a short time, or spread it all out in a long period of time. But it's not just leaves that show this. So I'm showing you a plot of the total number of heartbeats per lifetime in a mammal. Here's its body size, going from small organisms, here's a hamster and a rat, up to large body sizes. And so if you go through and calculate the total amount of life lived, in this case in terms of the total number of heartbeats, you come up with the answer 7.3 plus or minus 5.6 times 10 to the 8th. It's about 1 billion. You all have 1 billion heartbeats. I hope you're using them wisely. And those who are astute quantitatively will probably realize I've already used my 1 billion heartbeats. Don't worry, there is some variation in biology. Right? There does appear to be some variance, but in general, if you go from a small mammal to a large mammal, the number of heartbeats is about the same. Just like in leaves, right? But obviously no beating heart. So energy of life is constant across scale. So the amount of energy flux per unit mass is similar. During its lifetime, a mouse fluxes about a similar amount of energy per unit mass as an elephant. It's a mouse spinning away and an elephant slowly spending its metabolic capital. It turns out that it's about, to support a given amount of mass, it's about 8 times 10 to the 5th kilojoules per kilogram. So 8 times 10 to the 5th kilojoules per kilogram in plants and in animals. Why this number? Why an approximate constant? We don't know. So maybe we should step back and realize that other people have realized this too. So if we were to quote Shakespeare, pleasure and action make the hours seem short. So slow down, enjoy it. So to summarize, I've offered you 5 different insights. A scaling primer. I've given you some evidence that biological rates scale with changes in size, that biological times scale. I've focused on the importance of networks in biology, and that networks in biology seem to underlie the variation in biological times that we see. But ultimately, this variation in the scaling of biological rates and times are also constrained by these constants of life that we really don't understand. So, does biological time tick at a constant rate? Well, the answer appears to be no. Life's perception of time is different than geologic time, but biological time does have approximate constants. And we can reveal then these constants via scaling. So to conclude, what drives variation in biological time? Variation in the scaling of biological rates and times are ultimately controlled by the geometry of vascular networks that appear to reflect these generalities of life's design. And these approximate universal constraints on the total amount of energy that can be fluxed per mass per lifespan. So to go back to the oldest known human, approximately 122 years, and to go back to the longevity website, are we going to be able to extend human lifespan? Possibly. But I think these deeper underlying biological patterns are telling us something very important, not only about the quality of human life, but in terms of not only bettering the human condition. And if I were going to use one last quote, to quote Shakespeare, I've never known so young a body with so old a head. And so with that, there are many folks who have participated in this work and have inspired me, wonderful collaborators, as well as different funding sources and different institutional support. And so with that, thank you for your time and your interest, and I'm happy to take any questions.
Just a thought, and I'm not a biologist, so this could be complete rubbish. When you were talking about the fetus and the rate at which the fetus grows within the mum, you said that it grew at the mum's biological time rate, but when it's born, it starts to take on its rate as it grows. And obviously a fetus, for most people, is something they want inside them. A tumour isn't.
Yes.
So when many tumours grow, they grow at vastly accelerated rates compared with the rest of your body.
Yes.
Why is that? Is that network control?
So the question is, when a fetus is growing in your body, that's obviously something that you want, that is a growth that you want to have growing in your body, but also it is controlled, right? So mom is controlling the rate of growth, okay? And the metabolism is going as the rate of mom. But it actually turns out that one of the hallmarks of cancer is that within a tumor, effectively a cancer cell is a cell within the viewpoint of this theory is a cell that forgets that it's part of a network. And cancer cells, it turns out, tend to cluster their metabolism around then the metabolic rate of what you would expect then for a cell of a shrew. Right? And another hallmark of cancer is the redirection of vascular supply networks in order to support the energetic demands then of a growing cancer. But those individual cells, though, likely are not aware that they're a part of a growing and growing tumor. In fact, another reason why cancer can be so disastrous is that the inside then of a tumor becomes necrotic, okay, because a lot of those cells on the inside are not getting the supply that they need. Right? So it could very well be that cancer itself, at least some forms of cancer, are reflecting then this trade-off of cells forgetting that they're part then of a larger whole, a collective whole.
Thanks for the fascinating talk. I was thinking you didn't talk too much about temperature and its impact on metabolic rate. Often used to explain why, for example, creatures in Arctic regions grow really slowly. And so does that not throw off any of the constants that you were talking about? And also, to people who live in hotter places, presumably have a lower life expectancy, or people that do more exercise?
All right, so a lot of different questions then in there, but I'll take the first one. We'll see if we can get to the next one, the later ones afterwards. So your question first was on temperature, and I didn't talk about the importance of temperature. And so you would expect maybe temperature to be involved in here, because if you take, say, a given plant and you put it then in the refrigerator, and of course give it enough light so it can grow, but the cold then temperatures tend to slow then metabolism. Right? In warmer environments, you would expect metabolism to increase just with basic temperature kinetics. Right? Now mammals, we like to keep a constant homeostatic then temperature in order to maintain a very high metabolism. And in general, actually this work, everything that I presented, there's a whole temperature component of it. We can take these equations and put in temperature in this. And in fact, when we put in temperature, it improves our different predictions. So yes, in general, in colder than places, you would expect then metabolism to be slowed, which could then also impact then different life histories in terms of longevity then of things as well. Right? And so the temperature component is actually a very important other theoretical component, but I didn't have time to go into the details. But if you were quick, you noticed that I showed one plot where we standardized the growth rates of everything. We had to standardize the temperature at which their metabolism is running. So then you had another question associated with what we would expect then for people growing in hotter environments, whether or not we would expect differences in their physiology or diseases to be different. Is that right?
Right. Do heart rates speed up if we're in a hotter place? And therefore, does our lifespan decrease accordingly?
Do you pay the price for that? No, it's a great question, and the quick answer is that we don't know. But I do know one of my colleagues at the University of Arizona. He's an anthropologist, but he's a biological anthropologist, and he does a lot of putting people on treadmills to really changing their body temperatures, putting them in hot environments and cold environments. And his research is actually inspired from a lot of these scaling then ideas, and so he's actively working then on that. So I don't have a quick answer for you, but it's the type then, this sort of work then brings a different light to these sorts of questions. Yeah.
Thank you for a really nice talk. I really enjoyed it. I was wondering if in all the allometric regressions that you have shown today, when there's deviances, either positive or negative, if you can make predictions about whether those species may be likely to be endangered or become invasive if they are under higher or lower constraints.
Okay, so there a few different questions there. So the first having to do with the fact that because these plots are usually on logarithmic plots, right? We call them log-log plots. That we still have quite a bit of residual variation. And so in the case here, okay, we actually see that there's quite a bit of residual variation in amongst this line. Does that residual variation actually mean something important biologically, right? And can it be used, to inform kind of other biological questions? And the quick answer to both is yes, okay? What we tend to find is that this residual variation is due to things like temperature, but also due to the relative nutrient status, okay, or differences in life history of different organisms. So we actually find that there's a lot of biology then within this residual variation, right? And so once we control for the all-pervasive influence of size and then temperature, we can then begin to look at deviations then from kind of the mean response as an indication of what other factors in biology are important. Now, in terms of how these relationships can influence conservation, so if you think about an animal, ranging from small to large, in terms of recovery times, how long then it's going to take for a small animal versus a large animal then to recover. Larger animals need more time for their population densities then to recover. Okay? Excellent example of things like whales and whale recovery, how long it's taken us to recover populations then of whales. But it also means that for larger animals, that any sort of local extinction and reduction in the population densities, it's going to take us much longer to recover than those populations. And another thing that we're working on actually in Yadvinder's lab is that we're using these different scaling relationships to then understand how over the course of human history, one of the biological signatures of the Anthropocene is a gradual reduction in the maximum sizes of animals. Okay, so gradually we've killed off the megafauna, and increasingly we're reducing the population densities of larger and larger animals. And because of which, in terms of how long it will take to recover then those population densities, it's going to be quite longer, which is going to require a more intensive, but also many different lifespans of humans in order to make sure and maintain that conservation effort. Yeah.
Hi. You put a lot of your points with regard to scaling in the context of quite complex multicellular organisms.
Mm-hmm.
Now, you touched on protests quickly, but
Yes.
there's obviously a massive diversity at single cell organism
Mm-hmm.
level. How do these points carry over to those sort of contexts, and particularly in the context where you get sort of pseudo multicellularity arising, like in slime molds and things like that?
Yeah. So then the question is focused on what about metabolic rate scaling within unicells, in particular bacteria and some of the earliest of life. This is a very much an active and contentious area in the literature right now. One thing that appears to be the case is that once we get into unicells, there may be an evolutionary signal where we see these scaling relationships begin to congeal, okay, on some of the more recently derived unicellular clades, but also leading into multicellularity. So by the time we get to multicellulars, do we start to see these approximate 3-4 scaling relationships? And for some of these unicells, especially if we go the older than clades, what we tend to find is that these scaling relationships may not be as well behaved or as characterized, okay? Which is maybe what we would expect, okay, when the timing of natural selection and to effectively start operating on these internal vascular networks. But good question. Yes, Tim.
So how far away can you get from the lines if you're thinking about residuals? Sort of, can you get something that's incredibly slow in terms of, it's sort of
or incredibly fast for your size, right?
I'm wondering if you've identified the upper limit, which
Yeah.
you can't go above, but actually evolution can push things down way below.
Yeah. So it's perhaps not surprising that there is residual variation about this line, because I think that's biology, that's natural selection, trying then to break then these various different constraints. And depending on the local environment, maybe you can get away, okay, with not strictly obeying some of these different principles. And actually, one of my postdocs who's now in France, one thing that he's really interested is in crop plants, in particular using artificial selection to try to answer these sorts of questions, right? So when we artificially then select for increased production, increased growth rate effectively, do you basically break these scaling relationships? And don't want to kind of give everything away, but it looks as if many of these fundamental scaling relationships are still upheld, but in order to increase production, you have to basically manicure the environment to become effectively perfect. Okay? No competitors, no disease, resource input. Effectively, you have to increase the amount of energy from human societies in order to support these different crop plants that in no way, if you allowed them then to escape into the wild, would they ever last. So, excellent question. Yeah, in the back.
My understanding is that 300 million years ago, the atmosphere was much richer in oxygen, and therefore we had much larger, basically dinosaurs and everything like that. And so that's obviously that the oxygen level's not been constant over time, and also the sea has maybe 12% more oxygen in it, and therefore you can have larger animals in the sea because also the buoyancy of the water will support larger animals. So is oxygen also a factor?
Yeah. So I think the quick answer to the question is yes. All right, the oxygen concentrations appear to be quite important, and it appears to be correlated with maximum sizes that were around. Some good natural history examples actually come from insects. All right, so insects have similar kind of hierarchical branching tubes. Right. And they require then the movement then of muscles in order then to basically exchange then air from the outside then to the inside to effectively pump then the atmosphere from the outside to the inside. And oxygen is central then to aerobic respiration in many different organisms. And we find the emergence of these really large insects in particular in times of very high oxygen concentrations. And so some arguments have been that these high oxygen concentrations have enabled larger and larger body sizes then to evolve, which then suggests then that because metabolic rates have to be turned down so low as you increase then in size, that if you can increase oxygen concentrations, maybe you can get away then with increasing the efficiency then of supply and supporting larger and larger body sizes. But in many ways, this is still kind of basically back of the envelope calculations and ideas. But the concordance then between oxygen levels in the past and times at which we saw these very large organisms, in particular insects, seems to support that. Yeah. Well.
I'm a mere historian. I studied time in human consciousness. I studied time in human consciousness, and I've become aware that animals have their sense of time, their length of memory, what they regard as a long time or a short time. I was brought up on a farm, so I sort of watched the animals and what they expected and what they remembered and so on. Do the many-sided researches of your institute consider consciousness of time?
Consciousness of time.
In animals?
Well, it's an intriguing question. The quick answer is that within at least my department, a lot of my different colleagues, we haven't started to consider consciousness of time. But just as an anecdote, if I think back in my childhood and I think back about the passage of time, and my mother would say that, if you save up a given amount, of your allowance, in the end you can buy something really nice that you would like. And, that time span was on the, time span of about a year. Oh my goodness, to me that was forever, right? Waiting forever. Now a year passes like that. Now, does that inform us about consciousness and our perception, how biological time then intersects? I'm not sure. But I think that in talking then with people from different disciplines, in terms of how we can potentially merge, these different, studies of time in terms of the perception of time, I think would be fascinating. So,
thank you.
Yeah.
May I be permitted another one, please? A few months ago, here at the Oxford Martin, we heard a lecture about the deep sea in terms of the organisms that actually live and survive a couple of miles down in deep sea trenches. And the guy, if I remember rightly, said these were very, very ancient. They never see the sun. They don't get their power from the sun. They get it from the chemical reactions under the sea.
Sure.
Now, that would be a very, very interesting colony to study to see if that broke this rule, wouldn't it?
Yes, it would. All right, so you're talking about the deep sea vent organisms. These are ecosystems that are energetically run on a completely different set of biochemical reactions, and their energy ultimately comes from geothermal power, right, kind of within the Earth. I know of no detailed study trying to put those sorts of organisms on some of these different relationships. Part of the issue is that, once you You can't take these organisms in the deep sea and bring them up, to study in our lab because, they disintegrate and explode and so on because of the differences in pressure. And so it's quite hard to study their physiology, other than taking tissue samples and so on. But yeah, it would be fascinating. Yeah, absolutely fascinating.
Thank you, Brian. That was a fascinating lecture. I'd like to turn to the point you made at the end about these constants that emerge and what your hypotheses are of what these constants mean. Is it some aspect of biochemical machinery that just has so much wear and tear, you can run it fast or run it slow, but there's so many oscillations or heartbeats, mechanisms that you can run in a machine?
Right. So I was expecting this question probably from you, Yadvinder. So why these approximate constants? Well, I think ultimately it's entropy, right? And so life, with enough time does break down. And in order to maintain approximate homeostasis, in order to maintain then metabolic machinery in a state where not only resources are being delivered at close to the maximal rates, but then also that all of the biochemistry associated with growth and biosynthesis is maintained. So I can understand why it's this interplay of trying to maintain an internal homeostasis, but yet at the same time having the constraints of entropy, that with time things cannot last, ad infinitum. Why these exact numbers, I don't know. I have no idea. And I think that they would, form a very nice focal point to organize a lot of the biology that we study.
Great talk, Brian.
Thank you.
If we discover life on other planets, do you think it'll obey these constraints that you presented?
Well, I was going to be flippant with the deep sea question, but I decided to rein back. But since you've given me permission now to do so, so what if we did discover life on another planet? Okay, what if we did, if we were able to study their physiology, right? So I would predict that these constraints of taking in resources from the environment, converting them within a body in order to do work, right? That the principle of natural selection for any sort of replication and limiting resources is likely universal in the truest sense of the word, right? So if you have competition, limiting resources, I would think there would be a keen bit of selection to optimize resource uptake. And I wouldn't be surprised if quarter power scaling rules would be the result. And I've argued actually to astrobiologists that if they, by chance, ever got a chance to study life, that they should look for these sorts of signatures. Now, if there was only a quote-unquote quarter power signature that you could scan the stars with, maybe we could increase the speed at which we search for life. But yeah, but that's going way out on a limb, so, right.
That seems a suitably galactic point to finish your talk. So thank you, Brian, for that inspiring and broad-ranging talk as I really expect you to deliver. So I'd like us all to join and thank Brian for his wonderful talk.